Hi, everyone. I have the pleasure of introducing Amber Salzman of Adverum Biotechnology, who's going to give us a brief overview, and then we'll dive into Q&A.
Thank you, Steve. Okay. How do I move this? Oh, sorry. Of course, our safe harbor. So we're very privileged and excited to be in a really great position to advance therapies of deep meaning to patients. And the reason I say that is, firstly, we have really industry-leading platform capabilities. I think that just came up in the last session, where when it comes to gene therapy, the manufacturing of the vector sometimes is the gotcha, and I think that's where we do excel in process development. Also in terms of next generation vectors, the other thing that came up already, can you go to the cells of interest and transduce them in sufficient numbers so that you have efficacy? So those are two areas where we have leading industry capabilities, so we're very proud to have that platform. In addition to having the platform, we also have a robust pipeline. We have 3 programs moving towards the clinic. One of them is in wet age-related macular degeneration, and then the other 2 are liver-directed rare disease indications, alpha-1 antitrypsin deficiency and hereditary angioedema. We're also very fortunate to have the bank account to support moving forward with these programs through at least 2019. So that gives us a chance to really see if one of these lead indications, or potentially all 3, but we know the odds in this business, can have a meaningful impact on patients. We have a leadership team and organization in place to really bring these forward and have a tremendous impact in patient lives. So this is just visually our lead indications. As I mentioned, wet AMD, we came off of a previous program that did not have positive phase 2, and sometimes I feel like you learn a lot more from your failures than your successes. And based on that, we were able to bring forward a completely different molecule, where we have a different vector, a different transgene, a different promoter, and most excitingly, a different route of delivery. So we're going intravitreally instead of subretinal like the last program. That could be a real game changer for patients in this space. We also have the program for alpha-1 antitrypsin deficiency, where we have an open IND, and we will be moving into the clinic by the end of this year. Lastly, we have a program for hereditary angioedema. As many of you know, there's only one product on the market right now aimed at preventing these horrific attacks. And if you are on that medication, you still have breakthrough attacks. So we have the potential to help patients in that space as well, and we're moving forward in IND-enabling studies. In addition, it's worth noting that we also work with Editas and with Regeneron. I think those partnerships are very validating in the sense that if you're an organization that has expertise in the eye and you want to leverage gene therapy capabilities, we're the go-to company when it comes to having the vectors and having the capabilities to move those programs forward. So we're very pleased to be able to talk about them as our partners as well. So I'm going to stop there and let Steve quiz me.
Thanks, Amber. Maybe first on A1AT, maybe just talk about the approach, the vector, and contextualize the unmet need for us, given that there's an enzyme replacement therapy.
So right now there are 4 companies that market plasma-derived protein for this disease, which is a huge benefit over having nothing. However, the problem it poses to patients is that it requires IV infusions every single week. And that can cause a problem if you're trying to work, go somewhere, lead a normal life. And as a result, compliance becomes a problem. And because it's one of those diseases where you don't necessarily fall off the cliff the day after you've missed your treatment, it's just after a year you'll notice that you have trouble walking across the floor and walking across the room and still having enough lung capacity. So it kind of creeps up on you, and then you really regret missing that once weekly infusion. So here's an opportunity to be able to have a long-lasting treatment where your own body will express the protein, as opposed to having to go into the physician's office and present with progressive emphysema and early death.
In what routes of administration are you investigating?
So I will go back. You had asked me about the vector, and I guess it's worth noting that the reason we picked the serotype we did is there was a study comparing 25 different serotypes. It's published, and it showed that the serotype that we selected, the AVRH10, was the most efficacious in the lung. It's in a mouse for what it's worth, but there is rationale behind it. And then you were asking me, routes of delivery. So healthy people produce the vector in the liver. So clearly you'd want to have the liver produce enough of the protein to make its way into the lung because that's how it works for us. However, the thought process was that if you maybe delivered the vector into the pleural space, potentially you could also have your cells within the lung express the protein, and which is where it's needed, and therefore potentially with fewer cells being transduced, you could have enough of the protein that you needed, and hypothetically, you could potentially even go to higher doses. So with that in mind, we are going to look at both routes. We're going to look at IV, which I think has now, to say tradition in gene therapy is a little bit of an overstatement for this field, but traditionally when you're going to liver-directed gene therapy, the route of delivery is primarily intravenous. So we are going to look at the intravenous route to see if we can get sufficient protein expressed in the liver to make its way into the lung, but we will also be able to look at delivering it into the pleural space.
And what endpoints are you going to be looking at to evaluate which mechanism of administration to move forward with? What are you going to be looking at in that early trial?
Right. So, of course, it's a safety study, so we will primarily be looking at safety. But in terms of getting a read on efficacy, the 4 products that are on the market right now were all registered based on a biochemical endpoint. So if you can show that in the serum you hit a particular level, it happens to be 11 micromolars, that was what they registered on. So we have plans to engage with the FDA. This was an in-licensed product, and we have not yet talked to them about it, and we can further talk about that kind of efficacy endpoint with them.
And have you guys thought about whether or not you're going to use prophylactic steroids?
Yes, we have, and we are going to, when we talk to the FDA, talk about prophylactic steroids. We want to make sure that we give this program all the chances of success to see that we can get the levels we need. If we can then back off of steroids, we can consider it. But based on the data we've read out in the public domain, it seems advisable to start with the prophylactic steroids.
Moving on to some of the other pipeline programs, you described some of the differences on the wet AMD program from the prior AVA 101 approach. Maybe just overall, how much more potent and what is kind of the improvement you've seen in the animal models that are really giving you confidence that this approach can really overcome some of the limitations you saw before?
So there is clearly variability when you're doing the subretinal delivery. I mean, it's surgery, and there is variations, and depending on how sick the patient is or how diseased the eye is, you may not go as close underneath the fovea as if it was advanced disease. So it's a huge advantage to move away from that to intravitreal delivery. The reason why we're so excited about the preclinical data is there is an animal model where you go into, this is what's usually used when you're testing wet AMD therapies, where laser are put into the, you get a lesion based on lasers in the eye of a non-human primate, and then you look at if your therapy can clean up the neovascularization. So that's the way the model worked. So what we did is run some non-human primate studies. We used the CMV model, and we're able to show that our product worked just as well as Eylea did, and as well, in fact, as Lucentis did when we put the 2 different proteins in. So we're advancing our program where we're delivering aflibercept, and we have made public 20-week data, and at the 20-week, we still had consistently stable high protein expression. With the kind of promising results where it worked like standard of care, we've initiated long-term studies to make sure that the duration of treatment is in the time that we need it to be.
Moving on to HAE, hereditary angioedema and allergy. These aren't monogenic diseases. What led you to select these programs, and where do you think gene therapy fits into the treatment paradigm here?
So the reason why we were attracted to those programs is that they're all targeting the liver for protein expression. So as we move forward, the knowledge we gain from one indication can be applied to others. So if you look at A1AT, HAE allergy, they're all focused on the liver. So in that regard, there's learnings there. Also, in terms of the efficacy endpoints to see whether this is something that has meaningful data, in all of those programs, you can measure protein level in the circulation to see whether you're going to have a winner or not. So being able to have that kind of an early read, while there are incredibly exciting programs, for example, in neurodegenerative diseases, that have huge meaning to patients. Sometimes it's hard to see where the efficacy is because if it's slowly degenerating, it takes a long time to get an endpoint to see if you're having an impact. Some of the attraction we had was that we knew how to work with the liver, and we knew that if you could have early reads, you could work with that in a more meaningful way.
I think you touched on this in your presentation, but what do you consider to really be Adverum's kind of core competencies and expertise?
So I think we have a huge advantage, having had a program that we thought was going to move to phase 3. The company brought in experts and infrastructure that could support the process development required for this field. So I think what we've seen in the last couple of years is there's been some very exciting early data, and then there's a pause because if you're going systemically, you have to make a lot of this stuff. And some of the processes used to produce a small amount to get your early signal doesn't always scale. So I think it's a huge advantage that we are sitting on really industry-leading capabilities in process development. I think that's one side of the equation. The other thing is being able to develop the next generation of vectors. So we have a directed evolution approach that we're taking, as well as a rational design. And I'm sure you've heard there are other companies that do this, but where we do excel is in our ability to create libraries at the scale where we can screen them in non-human primates, which was a much more meaningful approach when you're looking for candidate vectors to meet the criteria that we're looking for. So that, along with the pipeline and really expertise and focus on the patient, I think puts us in a great place to have a huge impact in terms of meaningful therapies.
Any questions from the audience? I guess just last question. Maybe give us a quick snapshot of the Editas and Regeneron collaborations.
So in both of these cases, they're looking at making use of our proprietary vectors. As I mentioned on the wet AMD program, we have a serotype that works with intravitreal delivery into the eye, and that's fairly unique. And the data is very promising. So both of them wanted to look at having access to that vector to address diseases that they're going after. Also, in both cases, they really did look at us as a group of professionals that could aid them and guide them as they go forward and bring their technologies forward.
Great, thank you.
Thank you.